Okay. Assalamualaikum warahmatullahi wabarakatuh. Can everyone hear me?
Yes.
Yes.
Okay. Okay. Baik. Alhamdulillah. How many students we have?
29.
29. Okay. So we wait until we have 30 participants, and then start, inshallah. Because the class is going to be 3 hours. I don't know how the other lectures make for 3 hours lecture. Like Dr. Ridwan, he teach you 3 hours continuous, or he will stop like 1 hour and stop, and then there is another 1 hour, and then until finish the 3 hours? Or did he finish the what I call lecture like 1.5 hour and then dismiss?
He like continuous, like for 2 hours or so.
For 2 hours, so he gave lectures for 2 hours and not utilizing all the 3 hours lah. Am I correct?
Yes. Only Dr. Afendi did
Only
3 hours.
Only Dr. Effendi. Okay, only Dr. Effendi. Okay. If that is the case, let us wait for all the members to join us, inshallah. And then we shall start our discussion. Would that be okay, Shakira?
Yes.
Okay. So Shakira, by the way, you are in year what? Year 4, year 3, year I don't know.
Year 3.
Year 3. So biomedical is supposed to have 4 years, isn't it? So this is your 3rd year. Next year will be your last year.
Yes.
Okay. So maybe we can do a little bit of ta'aruf in the beginning of our discussion. Is that okay?
Inshallah. Can.
InsyaAllah. Okay. Saya ni orang Melayu juga, makan belacan semalam, makan apa tak tahu, tak ingat. So I hope that is okay if I mix with Malay, and if I for some terminology. But anyway, let us wait for all the members to join us, inshallah. So we hope that we will do this immunology, hematology, something that is fun. Saya pernah ajar kan sebelum ni? I have taught you before, isn't it? I'm not sure.
Yes.
Yes.
Okay, that's good. I don't know if we are face to face, maybe I can know who you are, but by looking into the name, because I have a big problem. I think I already mentioned to you in the beginning of my lecture previously. Memorizing name is quite difficult because I have too many students. Okay. There will be one student who's not going to join us. So this lecture will be recorded, and I hope that you are going to listen to this again and again. Okay. For this part, inshallah, we will have, since you are in the 3rd year, so I plan to do reverse classroom rather than having classroom where I teach all the time. I hope that for the next, for the other lecture, the other topics, I will listen from you. But I will give you some idea what are you going to do for the next lecture, because that's going to be another 3 hours, and that lecture will be this Friday. You still have plenty of time, and inshallah, I think that should be more effective rather than listening to me for a very long lecture. So I think can we start now, Shakira?
Yes.
Okay. Inshallah, as usual, let us begin the discussion today with the recitation of Kitab Al-Fatiha. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. الحمد لله رب العالمين الرحمن الرحيم مالك يوم الدين إياك نعبد وإياك نستعين اهدنا الصراط المستقيم صراط الذين أنعمت عليهم غير المغضوب عليهم ولا الضالين آمين. بسم الله الرحمن الرحيم. الحمد لله رب العالمين والصلاة والسلام على أشرف الأنبياء والمرسلين سيدنا محمد وعلى آله وصحبه أجمعين. اللهم افتح علينا حكمتك وانشر علينا من خزائن رحمتك يا أرحم الراحمين. رب اشرح لنا صدورنا ويسر لنا أمورنا واحلل عقدة من ألسنتنا يفقهوا أقوالنا. اللهم ألهمنا رشدي وعاذنا من شر نفسي. ربي زدنا عملنا وارزقنا فهما واسعا. Ya Allah Tuhan kami mohon ya Allah dengan rahmat dan kasih sayang ya Allah, ampunkanlah dosa kami, dosa kedua kepada guru kami, keberkahan berhak atas diri kami ya Allah. Ya Tuhan kami mohon ya Allah, terangkanlah hati-hati kami ya Allah, permudahkanlah bagi pelajar hamba ini Allah hari ini ya Allah, untuk faham apa benda yang hamba mu ajarkan ya Allah, dan berikanlah kefahaman yang sebenar ya Allah. Ya Allah Tuhan kami mohon ya Allah, jadikanlah ilmu ini ilmu yang bermanfaat dan jangan kau jadikan ilmu ini sebagai fitnah bagi diri kami. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzabannar. Alhamdulillah rabbil alamin. Okay. Right. Before we begin, as I said earlier, okay, let us do a little bit of again another ta'aruf. Okay. So this ta'aruf session, orang putih kata apa, tak kenal tak cinta, kan? Jadi saya pun nak kenal balik student saya ni, walaupun kita pernah jumpa dah sebelum ni. Cumanya bila saya tak tengok, when I didn't meet you face to face, I think it's quite not so, I mean, it's quite difficult for me to remember, especially we have met a few times, but we are dispersed between each other. I don't know which one is, yeah, I think I remember Nabahan. The brothers I have no problem, but the sisters I have a big problem. Kita panggil apa? Malu-malu gitu. Tak adalah. Alright, now I'm going to share my screen. Just before I'm sharing my screen, I think before I forgot, I am going to share with you my, what I call the attendance. This is very important because without the attendance, I'm afraid that you might not be able to enter the exam. Okay, so here is the attendance. I'm going to put it in the chat box. Okay, I would be very, very appreciate if you can sign in your attendance immediately. Don't have to wait until the end of the class. Sign in the attendance. You can do it now. I give you about 3 minutes to do it, and I will double check in the website. I attend. If there is enough 27, then we will have the lecture. We will have a discussion. We continue our discussion, Sean. So far I have 6. Okay, I have 25 students has been logged in. Can I have few more? I should have about 3 more. Okay, please log in the attendance. This is very, very important. Shakira, okay, you can hear me clearly, or is it like stop? It's not clear. You have like difficulties to hear me, to understand me. It's all good?
Kadang stuck, kadang tak. Jadi macam bila doktor kata stuck tu dia stuck.
Oh, tak boleh sebut kata stuck ni. Okay. Okay. If say for example, if you want me to repeat anything, just let me know, and then you can cut me in the middle of my lecture, because this is going to be a 3 hours lecture. But I try to I will finish it earlier because I have a meeting with Deputy Rector later on at 11. So I think I have to go make it short, inshallah. I hope that is okay for everyone. Okay. Now I have all the attendance, right? Now moving on to the topic that we want to discuss today. I noticed that the topic is supposed to be immunofluorescence, and another one is I don't remember what is it. Not electrophoresis. Okay, but that immunofluorescence and electrophoresis is quite straightforward. It's quite easy. That part I would like to pass to all of you. Later on, I will explain to you what should you do. Okay. It's quite straightforward, and I will divide you into few groups, and then each group will do a little bit of presentation. But before that, today we are going to focus on the topic which I love so much, because the name of the test. Okay, for the immunology we have various of tests. How do we know that the immune system, the antibody is there, the cytokines is there? Okay, it is not as simple as microorganisms. When you can observe it under the microscope, you can see how the structure looks like. What is the where is the location of the pili? Where is the location of the fimbriae? Where is the location of mitochondria? So on and so forth. But when it comes to immunology, you have to be a little bit more detailed. You are based on your hypothesis, assumption. Like we have to do like certain tagging to put that because tagging on the what I call the proteins, because you cannot easily observe the proteins unless you have to do something with it. You have to put on the gel, for example. Okay, why do we put on the gel? Why? And then there are also some people do study like putting on a paper. Okay, and then there are also study they put it on. As long as you can notice that there is a presence of the thing. Okay, then you are and then you do a little bit of confirmation, and you know that okay, that particular proteins or the particular antibodies are present in the aliquot. Okay, so now when it comes to the topic that I interest so much, the name is Eliza. I have my friend, named Eliza. I don't know why, but now she's a scientist already. She was my colleagues at University of Malaya. We both of us are quite lucky because during that time only two of us managed to graduate without extension because it's very, very common for the microbiologists to extend. Anyway, the thing is that Eliza is a very, very interesting what I call test, and a very straightforward, because what you need is the kit and 96 well plate reader or spectrophotometer. Okay, so you just need these two, and another thing is that the pipettes and pipette tubes, definitely. Okay, so you need only three things which will help you to run the ELISA. Another things that we are going to cover today will be flow cytometry. Okay, flow cytometry and ELISA. So what are the two things? Okay, I'm going to share with you some links from the YouTubes. I'm going to tell you some story about the flow cytometry from the ELISA, from my experience, because I've been doing this in the previous when I unfortunately, I don't know if there is any chance for you in the practical to do the two experiments. If there is any chance, then inshallah we'll do it. But if not, it's okay. At least you know the theory of it, you know how it works, and later on, inshallah, when you are entering into the industry, you will do it with your own hand. Okay. Now, the slide, I will provide you the slide later, but for now, I want to share with you this what we call the link. I want you to write down I put in the chat box the link to the Padlet. Okay. Okay, can you see my screen? ELISA and flow cytometry, the pinkish color, a bit greenish here.
Yes.
Can you see it? Okay. Now, I want you to go to the link. Okay, so padlet.com, Hafiz_Azmi/I love immunology. Before this, I didn't like immunology. I told you the story, isn't it? I don't like immunology so much because too many things I have to remember, and then too many structures, too many mechanisms, and so on and so forth. But if we make it something like a story, then inshallah it will be very, very interesting. Okay, now I want you, I hope that you are now with me on the Padlet. But in case you don't have any other device, you are currently using your phone, you only have your phone, it's okay. But the rest, if you are able to join the Padlet, that will be super excellent. Okay, now let's do the activity. Okay, what I want you to do is simple. Okay, there's a add button. Okay, and then with the add button, okay, you can like add something. You can write like your name. Okay, I put my name, Hafiz. Okay, and then I want to write about, and then I want you to tell me, what do you understand about Eliza? Okay, when I said Eliza, what comes into your mind? Like for me, I remember my friend. I think when I talk about Eliza, my friend, I also think about Eliza is the antibody testing. Okay. Now, and then click publish, and it will appear on the dashboard. Okay, what I want you to do is very simple. Same like what I did. Write your name. What do you want me to call you? Okay, and then when I mentioned Eliza earlier, what comes into your mind? Okay, now I would like to give you 3 minutes. Okay, help me to write down whatever thing comes into your mind when I first mentioned about Eliza. Okay, 3 minutes from. I have to quit my WhatsApp. Otherwise, it's going to disturb you. Okay. If you have any problem, all right. Fatini says, okay. Fatini says Eliza is a machine. Okay. What else? Remember, you are going to become you are already a biomedical student. You are a biomedical biomedicine. Okay. You are already a scientist. Okay. It's a test that what we use. Okay. What else? Okay, I have Shakira here. Shakira, thank you, Shakira. What? Okay, other members, we have got 27 participants, 27 students. Right. Now, Nadia says, laboratory. Oh, laju. Okay, laboratory. Mana dia? Okay, so Nadia, okay, Nadia, Eliza, a biochemistry essay, enzyme link. Okay, so meaning that something that involves enzymes, perhaps. Okay. Fadilah say it is an immunoassay. Immunoassay. Okay. Eliza, biochemical test. Eliza, biochemistry essay, a kit for a quick test research tool. Okay, Farhana said that. And then I have here Nadia with Y. Eliza, something to do with science test, has full name enzyme link immunoabsorbent assay. Okay, we have Akilah biochemical test. So we have about 17 answer already. Fadilah. Okay, Eliza biochemistry assay. Eliza, easy to use immuno test. All right. We have 18. Nadia Sharifah, a test involved in immunology. Masturina. Oh, it makes you remember Dr. Ridwan, the microplate reader. Okay, perfect. So we have Sahira, easy to use. Okay. Now, Hayati said antigen diagnosed disease. All right. Now, every one of us know what is the function of ELISA. By default, we know why we use ELISA and what are the machine that we need to run ELISA. We need the microplate. Okay, and we also know that ELISA is a kit. It comes with a kit. Okay. Mizan said it is ELISA, something to do with laboratory. It is a test antibody, and it tests the antibodies in the blood. Okay. Now, Nadia, okay, this one has full name. This is done. Right. Now, every one of us know, okay, what is ELISA? By default, we know lah, the ELISA is something that is involving the machine. Okay, it involves immunology tests. It involves biochemistry tests. Okay, anyone would like to disagree with any of the answer given by your friend here? ELISA is used to detect antibodies in blood using laboratory tests. Anyone would like to say, sorry, doctor, I agree. I think I disagree with Mizan, for example. I disagree with Farhana. I think that is not a laboratory test. Anyone would like to say something, or you want to listen to my explanation first? Okay, I stop sharing on the Padlet. Stop sharing. Okay. Okay, Najia. So what is your answer earlier when we talk about ELISA?
Test is that is quite common sebab pernah dengar sebelum ni dalam like.
Okay, so ELISA is a very common, kan? It's a very common test, and people always do that. Okay, but the thing is that you are going to become an individual that is going to enter the laboratory, okay, to do some testing, okay, to verify whatever doctor said, okay, that doctor request from you some data analysis, okay, to confirm whether the antibodies exist or not. Now, my another question, right? So this is a little bit of critical thinking, based on what you have learned previously, right? Now we are talking about this ELISA, as what you have mentioned. We have learned about ELISA. So you think about something involves antibody. Now, in the current situation of COVID-19, is it possible to do a detection of someone's having COVID or not having COVID using ELISA? What do you think, Najia? Is it possible to use ELISA for testing the presence of COVID-19 in certain individual? What do you think, Najia? Sudah, Dr. Hafiz ni tanya soalan semua dia ni. So this is how I know whether you are really interesting to know more about ELISA or not, or if you are having something in your mind, you know, before we discuss, and then we will discuss around that, inshallah. Okay, Najia, what do you think? Can you replace? Yeah.
No.
No. Why? Why do you think cannot? Or would you like to ask someone else to give the answer?
Is it because after vaccine the antibody will develop? So the result will be macam lain ke? Lupa.
Now the question is, can you detect or not? Okay, how about Mizan? What do you think, Mizan? Can you detect someone's having COVID by using ELISA?
Yes.
Yes. Your justification? Okay, remember what we have learned before in our, I don't know which year, we talked about immunology, what I call the basic immunology, but try to remember that a bit as well when we talk about the innate, adaptive, and so on. If you say yes, why?
Because we detect the immunoglobulin in the blood. For example, IgG.
Okay. Okay, Wizan, thank you very much. So you detect the antibody. So we know that there is an antibody that is specific towards COVID, for example, and then we design an antibody that is specific towards it. If there is any attachment, later on we can say that, okay, there's a presence of COVID-19. But doctor, I'm not so clear. You don't have anything. You don't share anything. You don't even share your screen. Okay, now, by understanding the concept which I just mentioned to you earlier, okay, moving on to my slide. All right. Okay. Remember not to worry about your exam, because your exam, definitely I will bring it from the slide. But now the objective, my objective to teach you, to share with you here today, I want you to be very, very clear in whatever research or whatever experiment you do, there must be an objective of it. Okay. Why don't we do ELISA? Why we just go for PCR? Why is it has to be PCR for the what I call as the ultimate test to detect COVID-19? Okay, and why do we use saliva kit? Okay, so you know saliva kit, and then it gives you certain result. Does the saliva kit as a part is considered as an ELISA? Because by listening to this lecture later on, you will understand and you will think about it, whether the saliva kit that we are currently using, is it a part of ELISA or it is not? Okay. With PT test to detect someone's having pregnancy or not. So there is also could be, you know, the concept could be similar to ELISA or maybe not. Okay, but that is the objective of my sharing session for today. Okay, it is not just for the sake of your exam. Okay, it is more than that. Okay, give me a second. I would like I want to. Okay. Okay, intern Rahana, so far so good?
Yes.
Okay. So again, objektif kita untuk faham. Okay. Hafal tu nanti lupa lah. Okay. Remember when, inshallah, when you are entering like path lab later on, or maybe in the hospital, okay, when you run this ELISA and flow cytometry, we are going to discuss on flow cytometry too later. Okay. You have to remember, not to remember me, but to remember the concept. Why are we doing ELISA? Okay, is it because ELISA is more expensive compared to PCR? Is it because of PCR is far more reliable, far more, you know, the detection is very high, very quick, very, very precise. Maybe the concept of detection is different. So these are the things that you have to put into consideration. Now moving on to the slide that I'm going to discuss today. Okay. Now, what I want you to have at the end of this lecture, okay, we are going to discuss on two things today. It's quite a big topic. Okay, we want to talk about ELISA. We want to understand the principle. Okay, why we do ELISA, and also we want to understand what is the principle of flow cytometry. Okay, say for example, I give an example. When I'm talking about principle, you have here the letter M. I don't know if you can see on my camera. I have M here in front of me. It is made of wood. Okay, it is made of wood. Okay, now if I want to cut this letter M that is made from wood, can I use scissors or should I use knife to cut it? Okay, if you use scissors, you know the reason, the principle of scissors. The principle of both of them, the principles is to do cutting. Okay, but which one is more suitable to cut this woody letter M? Okay, should I cut with scissors or should I cut with the knife? Okay, so that is the same thing here. I want you to know when you want to do in the lab later on, if you want to advise in the lab, Okay, if you have your own lab, okay, I hope that one day you will have your own lab. You will create your own path lab. Who knows? Inshallah, I hope that is going to happen. Become an entrepreneur, right? Then you will understand why should I choose Eliza, because Eliza kit is very expensive. 96 well plate, you can only run about 50 tests, and for each 50 tests, it costs you about 1,500 for one plate for detection of IgA alone, for example. Okay, 1,500 ringgit. And then of this 1,500 ringgit, that is only for the kit. How about you to pay your staff? How about the consumables, like to do the service? These are the things that you have to put in consideration. That's why you have to understand, really understand the principle of ELISA as well as flow cytometry. Okay, we will talk about flow cytometry later. Now I'm focusing on ELISA. Now, number two, you should also understand the concept of antigen-antibody interaction. You know, antigen must be very, very specifically attached to the specific antibody. You have a specific antibody against specific antigen. Okay, so if you have antigen A, it has to bind with the antibody A. If it is antigen B, you put antibody A, there will be no reaction. So that is the thing that you have to understand. You should be able to link this concept. Right? Last but not least, you should be able to explain what is the function of each of the tests that we are going to discuss today, both ELISA and flow cytometry. Okay, Mizan Kamalina? Boleh? Now, moving on to the next slide. Okay, now we are in the next journey of understanding, remembering what is the antibodies. We have learned about this. Okay, now, this is a very basic. We know that when we talk about antibodies, it's something that can be found in the body, okay, in our body, in the blood. Okay, we know that this antibody is produced by B cell. We know that this antibody is also something that is very specific to it, antigen. If it is antigen, say for example, SARS-CoV-2, okay, it will bind only to SARS-CoV-2. If it is the antigen, like for example, if you put Candida, and it will only bind to the Candida. If it is going to be very, very specific, okay. Say for example, if you put the antibody specifically targeting the what they call the spike protein. Say for example, I give a spike protein name Hafiz spike protein. That it will, if you have a specific antigen that antibody that binds to the antigen Hafiz spike protein, then the antigen has to be very, very specific. It will bind, okay, the antibody will be very, very specific that bind to that spike protein of Hafiz spike protein. Yeah? So it has to be very, very specific. If there is a slight mutation, then this antibody will not bind anymore. All right. So you have to remember that the concept of mutation, the concept of antibodies, the concept of antigen, even in this study. Now, we know that when we talk about antibodies, there is antigen binding site, and we have variable constant. Okay, we have light and heavy chain. All right. What is the most important is this bit. Okay, the site where the antibody can bind to the specific antigen. All right. This is also something that is very much what you have learned previously. So when we talk about antibody, how does this product produced? Right? We know that when we talk about antibody, okay, in human, we have the origin from, okay, which part is the original, the origins of the B cell? Okay, what are the organ that produce B cell? Can I have Azza to tell me what is the organ that produce B cell? Try to remember your lecture on immunotolerance if you have any before. Najia, can you tell me where is the original location of, or I would say, the organ that produce B cell? What is the organ that produces B cell? Sharifah, Sharif?
Bone marrow.
Betul lah tu. Okay. So we have bone marrow. Okay, so this bone marrow, okay, not only exists in human, it also exists in other mammals, such as mouse, rat, rabbit, goat, sheep, horse. Okay, as long as mammals lah. Okay, because we cannot produce the antibody from human if we want to run experiment. Is that correct, Shafiqa? How many blood that you want to take out from human just to produce this antibody if you want to do it for the sake of an experiment? It's unethical. Do you agree with me, Hanis? It is unethical, isn't it? If you want to produce, to take the antibody from human for the sake of for your experiment. Okay, now the question is the ethics. Because of that reason, okay, there is a must to have an alternative to produce these antibodies. Rather than using human, we go for mammals. Okay, we go for mouse, we go for rats, rabbits, and so on. Okay. Yeah. So when you receive a kit, ELISA kit, then you know the antibody that comes together. Okay. Normally it comes from these animals. Okay, so the antibody comes from these animals. Okay, next, this blood, okay, the blood that is extracted from these animals, the mouse, rat, rabbits, and so on and so forth, okay, it contains polyclonal antibodies, which is a multiple antibodies that can bind to the same antigen. Okay, where is the location of the antibody? It will be in the serum, okay, which now we call this antibody as antiserum. Okay, so now you understand what is the meaning of antiserum, right? Let's move on to the second definition. What is the antigens? Okay, this is what you have learned before. When we talk about immunology, there must be an antigen. So an antigen is something, a substance that stimulates, okay, that stimulates the production of the antibody. Say, for example, if I put bacteria in you, so the bacteria will be the antigen. If I put, say, for example, someone having implant, and the implant is something that is rejected by the body. So the body recognizes this implant as an antigen, even though it is not a microbe. Something that is not naturally exists in the beginning of the, the naturally produced by the body. Okay, so it antigen is present naturally in the body, like hormones. Hormones is one of the antigen. Okay, it's not present in the body in normal condition like drugs. Okay, next, when we talk about immunoassay, like what we have some of our sisters here, Fadilah mentioned, saying, oh, Dr. Eliza is an immunoassay. Yes, it is an immunoassay. Immunoassay, for your understanding, it is a test. Assay means experiment, study, a test. Okay, so this is a test in the laboratory which use the concept of binding of antigen, okay, and its homologous antibody. Okay, it binds the antigen with the homologous antibody to identify and to quantify. Okay, you can identify, you can even quantify how much antibody, how much antigen in the sample. Okay, Shafiqa, so far so good? Okay, boring dah, ngantuk dah. apa nama, Syafiqah Yusriha, okay
Doctor, I have one question
Yes, yes.
regarding the antigen.
Yes.
The second point is present naturally in the body like hormone. How this how this statement apa ni masukkan apa yang
Okay.
point ni masukkan. Boleh Doktor?
Boleh.
Masih Doktor.
Okay. There are many things exist in our body. Okay, in the blood we have the antibodies, we have proteins and so on and so forth. Okay, among the proteins, okay, that is considered as a foreign body, or okay, foreign body is an antigen, as something that is classified as non-self, is the hormone. Okay, maksud kata, okay, antigen seperti contoh antigen ni biasanya most of the time we thought that antigen is something that comes from outside. Okay, we always thought that antigen like bacteria is an antigen, fungus, virus and so on as an antigen. But there is also an antigen, okay, that is produced in our body by our own system. Okay, so this antigen is naturally produced, and the antigen, one of the antigen that is naturally produced in our body is this protein, or what we call as the hormone. Okay, Nabhan, clear? Do you understand? Can you get me?
Yes, Doctor. Alhamdulillah.
Okay, Alhamdulillah. Okay, because this lecture is going to be 3 hours, I'm so sorry if my pace is very slow, but let me know if it is too slow, then I will make it very quick. Okay, so far so good, eh? Okay, now moving on to the next what I call terminology. Now, when we talk about this Eliza again, so we have what is a term what we call as the analyte. Okay, so when we talk about analyte is something that is very much relevant to, which is very similar to analyze. Okay, so analyte is the sample, okay, that you are going to analyze. Okay, in immunoassay, the analyte is either antibody or antigen. Okay, remember when you come into the lab, okay, if someone say, okay, what is the analyte? Then you can answer, okay, the analyte is antibody, or the analyte is antigen. That is the sample that you want to test. Okay, so it's analyte. Okay, you cannot. You are now a scientist. Okay, you're already in 3rd year, so I consider you as a junior scientist now. Now, when you become a scientist, then you have to understand. When you talk about antigen, what is antigen? You cannot. By the time now you are in the 3rd year, okay, you should be able to differentiate between all this. What is analyte? What is antibody? What is antigen? Okay, you cannot confuse. You know, like it should be on your fingertips. Okay. Like I'm in dentistry, I have to know in my 3rd year, I should know what is incisor, what is the apa gigi geraham nama apa, what is the numbering and so on and so forth. So that is also the same with you. When we talk about analyte, then you have immediately comes into your mind, okay, analyte, okay, what is the analyte? Then you can easily understand, okay, I want okay to test the antibody, I want to test the antigen. Okay, now that is the terminology. Okay, this is like what I just mentioned to you earlier. Antigen must be very specific to the antibody. Okay, if the antigen looks this shape, it definitely is not the shape lah. Okay, I took it from the textbook. Okay, you can have the textbook. I already put everything in my website. Okay, just type hafizalmi.com and that will be there. I think the lecture note is also there. All right. So you have this antigen. Okay, it will bind to the specific antibody. That is why when I have a debate when it comes to the development of because I am part of the vaccine Muhammad, or what we call that under the MOSTI, vaccine communication team by Young Scientist Network, Academy Science of Malaysia, Minister of Science Technology. So in that discussion, we discussed, okay, like we have alpha, beta, gamma, and so on and so forth, the mutation of the virus, COVID-19. So now we are producing the antibody with the COVID-19 vaccine that we have so far. It is very specific to this, say for example, to this antigen, to this side chain. Now, if I change this side chain, but I didn't change this antibody, would it be okay? Is it still going to give some, positive data? So we have a very big discussion on that, but I don't want to discuss about that in this lecture because we are talking about the technology. But you have to think about this. If you want to do ELISA test, for example, for certain antibody, okay, then you have to remember the antigen must be very, very similar, 100% similar to the antibody that you have designed. Okay, if you change a little bit, like this one yellow, you change just like this kind of shape, even though it looks similar, but it's different based on the antibody antigen recognition, and it will not bind, it will not give you any result. Okay, remember, you are an expert in biomedical. You know better than the others. Okay, so you have to understand the concept. If the results does not appear, it could be the antibody is not specific enough to the antigen, or the antigen that you put is not related or relevant to the antibody of interest, right? Now, as a lab personnel, okay, now I would say that nambahan, say for example, you are the owner of path lab, okay, so you want everything to be perfect, clear, reliable. When you give the results to the hospital will not blame, will not query any of your technique. Okay, do you agree with me, nambahan?
Yes, okay, yes.
Yes, that is the ultimate when we develop a company. Okay, then especially when we talk about a company of like Path Lab or some other company like biomedical company to many others. Okay, then perhaps you want to expand your company to ensure that all the hospital facilities will send to you to test the what do you call that their sample, or even if you want to develop your own kit, okay, like what I'm currently doing, okay. So I want to develop my own kit to detect caries. We know the caries is going to happen, but we want to make it a very, very early, maybe in the beginning of the age of the infants, before the once the tooth erupt, we want to detect as quick as that. Okay, when caries will start, what should we do to prevent the caries? But anyway, that is another technology. But now we are talking about the technology that already exists many years ago. Okay, this is the ELISA technique. Okay, more than 20 years, 30, 40 years, I think it's a very old technique. All right, but still useful. Okay, biochemical technique, like what we have our sister here, Nadia said. Okay, it's a biochemistry essay. Okay, we have a few brothers, a few sisters saying the same thing. Okay, it's a biochemistry essay. That is correct. Okay, Akila. Well done, Akila. So it is a biochemistry test. Akila ada, Akila? Mana Akila? Tak dengar Akila suara. Boring saya cakap seorang-seorang. Letak dalam kelas. Okay. So Akila and all the members. So this is a biochemical test. All right. So this biochemical test is a combination of biochemistry and immunology. Okay. So it is mainly the purpose is to detect the presence of antibody and antigen in your sample. So the sample can be various. Okay. The sample can be blood, can be sputum, can be saliva, can be urine, can be stool, but you have to dilute it. Can be anything. Okay, as long as you can make it in the liquid. Okay, right. Now, so we have various types of ELISA. But in this lecture, I just want to focus 3 types of ELISA. Okay, so the first types of ELISA, we name it as indirect ELISA. Okay, indirect ELISA. The second one, okay, I'm sorry those who are fasting. So the second one is known as sandwich ELISA. Okay, sandwich ELISA. Okay, some people call it as direct ELISA. So we have indirect, direct, and the last one is competitive ELISA. Okay, there are a few more types of ELISA, but in this class, I just want to focus on these 3. Okay, fine. So remember, indirect, sandwich, competitive. Indirect, sandwich. Sandwich is easier for you to remember based on the understand. It will be more easier for you to understand. Okay, rather than calling it as a direct. Okay. Now we go for the first one, indirect ELISA. Okay. So when we talk about indirect ELISA, what we do is that, okay, in the laboratory, okay, the kit comes to you, the kit that provided to you will already have an antigen coated well. Okay, the well that is already coated with the antigen. Okay. Number 2, what will you do is that you have to wash the, you are going to remove what I call the suspension, okay, the supernatant, okay, and then you will add specific antibody to be measured. Okay, you add specific antibody to be measured. All right, the next one, you remove the aliquot again, okay, and then you add conjugated secondary antibody. This conjugated secondary antibody is specific to the antibody that is specific to the antibody that you put, okay, in the second step. Okay, and then this secondary antibody, it has the enzyme attached to it. Okay, this E is an enzyme. Okay, so imagine when you remove the supernatant, you add on a substrate. If the substrate, okay, not if now, okay, so this substrate will be cut by the enzyme and change this into fluoresce. Okay, it gives fluorescent, it gives colors. Okay, so that's why when you do ELISA, indirect ELISA, okay, if the antibody here is specific to the antigen code, then you will have the what I call the secondary antibody will also bind to the antibody that is specific to the antigen code, and then eventually it will give the fluorescent color when the substrate is cut by the enzyme that is already attached to the secondary antibody. Okay, to make it very simple for you to understand, say for example, you have the antigen coated well that comes to you in the kit, you wash it, but you have a sample where the antibody is not specific to the coated antigen. What will happen? It will always remain empty. Okay, it will remain empty as antigen coated well, because the sample here is not binding to the antigen that already coat on the well surface. If there is no where is the annotate? Okay, look at this. If there is no specific antibody from the sample attached to this, the question is, can secondary antibody bind to this red bits, the antigen? The answer is no, because this secondary antibody is specific to this sample. Okay, so when there is no binding of the secondary antibody, enzyme conjugated secondary antibody, you put the substrate. Okay, you pour in the substrate. A color is there any fluorescence? Can you detect anything? If nothing here, you put the substrate in. Akila? Yes. Anyone who would like to answer? Okay, Akila.
Yes. No.
No. Okay. So there will be no color change, because the secondary antibody is not present. Okay. So that is, say for example, if I ask you in the exam, okay, I put here antibody code for COVID-19. Okay, but you have a sample from the patient, okay, of negative COVID. You put inside here, okay, you put the secondary antibody. If it is negative, would it be yellow color, the fluorescent yellow color or not? So the answer will be no. Okay, so that is such of the question that I will give you if I want to give. Okay, yeah. Now you have to understand what is this indirect Eliza. So it starts from the antigen coated in the well. Okay, antigen coated in the well, that occurs first. Okay, now I have to, it's okay, I just move to the next one, we'll delete later. All right. Yeah, all right, that's true, I have to delete all the annotate. Annotate, how do I delete this? Yeah, I have to delete this. All right. Now, the protein antigen to be tested for is added to each well of ELISA plate, okay, where it is given time to adhere to the plastic through charge interaction. Okay, a solution of non-reacting protein is added to block any plastic surface in the well that remains uncoated by the protein antigen. Okay, so this antigen is already coated. Okay, okay, I do a little bit of mistake. So this is the antigen that comes from the patient. This is the antibody. Okay. This is the method. A solution of non-reacting protein, okay, is added to block any plastic surface in the well that remains uncoated by the protein antigen. Then the serum is added. This is the what I call the general method lah. Okay, if I put the blank, for example, then you will understand. I make an exam question and I put blank, and then you can fill in the blank if there is any such possible to have that kind of question. Okay, so then you put the serum. Okay, so this serum contains a mixture of the serum antibody, okay, of unknown concentration, some of which may bind specifically to the test antigen that is coating the well. Okay, so the serum is added, which contains a mixture of serum antibodies of unknown concentration. You don't know what is the concentration. Okay, so afterward, a secondary antibody is added, which I just mentioned to you earlier. All right. Okay, so later on, you add on the substrate for this enzyme, and the substrate will change the color. All right. And then when there is a presence of the antigen, then you will immediately say that, okay, now I know there is a presence of this what I call antigen. Then there is potential. Then there is a potential of these individuals having certain diseases of what causing them the infection. Okay, the higher intensity of the color, okay, which I showed to you in this figure. Okay, the yellowish it is. Okay, the higher number. Okay, the more quantity of the antibody. Okay, the higher quantity of the antigen. Okay, the higher quantity of the antigen from the samples that you took. All right. Now, make a okay. This is another summary. Okay, for the first method, the indirect ELISA, you have the antigen bind on the surface. Okay, antigen, you take it from the patient, bind on the surface. You to the substrate, to the antigen, it will bind to it. Okay, once it binds, you put the secondary antibody that is specific towards the first antibody. Okay, this secondary antibody has its fluoresce, has its enzyme. Okay, and when you have the secondary antibody bind to the first antibody, the primary antibody, okay, you put the substrate, it will give you color. Okay, so in this figure, it gives you pink color. Okay, it depends. Certain kit gives you different color. Some gives pink color, some gives the picture I showed to you, yellow color. Okay, so this one is bluish color or purple color. Okay, so far so good, Masturina?
Yes, doctor.
Okay, if anyone confused, let me know. Okay, if anyone confused, just stop me. Okay, I will make you clear before we go to the next one. All right, now moving on. Let me ask first lah. Raudah, okay, Raudah?
Yes, doctor.
Okay. Baik. I was confused about this before when I did my first degree, and then I have to read many times. And when I asked my professor that time, my professor said, Hafiz, this is very simple. How can you don't understand? And from that, I start to have like, okay, if it is so simple, then maybe I am the one who are not good. But now, whether you are good or not good, it is not about you. It is not your problem. It is about me who are not able to teach you, to share with you properly. Okay? Don't have to put blame on you if you don't understand. Now, I already asked you a few questions. If you don't understand, that is your problem. If you have any problems, again, just keep on asking me. Inshallah, I will answer to you. That is my responsibility. Now, moving on. Yeah.
Sorry, saya ada soalan. Regarding gambar the well tu kan.
Which well? This one?
Yeah. Kenapa dia punya color lain-lain ni? Dia punya
okay. Color lain-lain. Soalan yang baik. Baik. Satu well, one well for one sample. Okay, over here are all the control. Okay, so there will be also a standard. Okay, standard meaning that it shows like 100 microgram of antibody. Okay, so if you have this color, you compare to this color. Okay, you compare it through the reading of spectrophotometer. Then the machine will predict. Okay, if this is the intensity, then it is actually like certain picogram. This is going to be less and less picogram. So this is diluted. Okay, so another thing is that you have to remember. Okay, each well represents different sample. Okay, each well you can put different sample in different well. Okay, so for example, if you have 96 well plate, you have 40 samples, then you can load all the 40 samples in one plate. So this is what I call, I took this picture, I don't remember where I took this picture, maybe from a website somewhere. So it's actually 96. So here, if you count 4 times 2, 4, 6, 8, 9, 12. 12 times 4. It's actually not 12 times 4. Okay, it's 9612. Okay. The digit, there are a few more at the bottom. Right? Now, I hope that answer your question. Ni sampel lain-lain. Okay.
Okay, thank you, Doctor.
Okay. So done with panggil apa tadi, indirect. Now we move on to the sandwich. Because sometimes you are confused whether to call it direct or indirect. You put indirect, become direct, direct becomes indirect. So you put the name sandwich, immediately you can get rid your confusion on whether to put it direct or indirect. Okay. Now, okay, the ELISA plate is coated with antibody, okay, to detect specific antigen. So in this case, okay, you put the antibody, okay, the plate comes to you, okay, with antibody on it. Okay, the plate comes to you with the antibody on it. All right. So what will happen is that when you have the antibody, you put the sample from saliva, for example, okay, and then say, for example, in that saliva possess the antigen that is specific to the antibody down here. Okay, the antigen will immediately bind to the primary antibody. This is also known as a primary antibody. Later on, what you are going to do is that you are going to add the secondary antibody. Okay, and the story will continue later lah. Right now, the difference between sandwich and indirect ELISA is that the sandwich ELISA, okay, it already has its primary antibody attached to the sandwich, to the surface of the well. Okay, compared to the previous one, it is antigen that is attached to the surface. Okay, this one, antibody attached to the surface. Okay, Radah, can you see the difference?
Yes, Doctor.
Okay. Now, the story on the sandwich is quite similar. The only difference is that what comes first? Okay, when antibody comes first, okay, so this is the antibody coated wells. It comes with the coated antibody. Okay, you wash it, you put the antigen. Okay, from the saliva, you put the antigen. If the antigen present that is specific to the antibody of your interest. So, for example, this is antibody specific for COVID-19. You take the saliva, spit, you put you have the COVID bind on it. Okay, and then you remove the supernatant. Supernatant is your suspension lah. Okay, and then you wash it. Okay, and then you add the secondary antibody. Okay, the secondary antibody is an enzyme conjugated secondary antibody. So you have the secondary antibody binds to it. All right, and this secondary antibody has its own enzyme. You wash it, you put the substrate, and poof, it changes the color. Right, it gives a fluorescent color. It depends, So this is a substrate, and it will be reacted with the enzyme and gives you a certain color. And different color will give different concentration of antigen will give you different color or different intensity of your color. Okay, so remember the difference between the two. Sandwich starts with antibody. Okay, this is the more details on what I have mentioned to you. Okay, so you have to prepare the surface to which a known quantity of captured antibodies bound, and then you block any non-specific binding sites. Okay, you block after you block them, and then you will apply antigen containing sample. You put a sample, okay, and then you wash it, okay, the unbound antigen, and then you add what I call the secondary antibody. Okay, apply enzyme link primary antibody detection antigen. All right, here I put it a secondary. Okay, So it should be the secondary, yeah? Right. So you put the link to maybe, okay. So you put the secondary antibody, all right? And then you wash the plate, okay? And you remove those antibody enzyme conjugated that is not bind to the antigen, and then similarly you check the absorbance. This is of it. Sample attached to the solid surface bind primary, wash, label with the secondary antibody, and eventually you read and you get the result. Okay, Sharifah, Sharif, so far so good?
Yes, doctor.
Okay, clear, Alhamdulillah. Now, the next one, competitive. Competitive nama pun compete. Okay, so this is a competition. Competitive ELISA in other, it's almost similar to what we have been discussing earlier. Okay, the only difference is that what starts, okay, okay, in the beginning of the test. Okay, so for this competitive, okay, we incubate antibody with antigen. Okay, antibody that comes with the antigen. Okay, that we want to measure. Okay, we put the antibody with the antigen. Okay, so in the mixture, so add antigen, antibody mixture to antigen coated well. So this one is firstly coated by the antigen. Okay, so what will happen is that the antibody over here will bind to the antigen coated well, right? Those antigen that already bind to the antibody, can it bind again to the antigen that is coated? No, it is not. Then what will happen then, we put the secondary antibody, the same thing happens, substrate, specific substrate, and eventually we'll have the fluorescent color. So this is the concept of the competitive. The antigen and the antibody, okay, the antigen and the antibody, okay, it's already okay, it's already bind, okay, the antigen and the antibody is already bind in the beginning. All right, they just put together with the another antibody that is not bind to any antigen. All right, and you can see there is a competition occurred here, and eventually you give, it still gives you the same content. Right, so for this method, okay, the label antigen competes for primary antibody, the label antigen. So this label antigen, the one that is competing with the antibody with the sample that is unlabeled. Okay, so the more antigen in the sample, the less labeled antigen is retained. Okay, remember, the more antigen in the sample, so you will have less antigen is retained, and the weaker signal you will have. Okay, so this is what I call that, the the label antigen. Okay, so we have more antigen, label antigen will retain, okay, and then you will have weak signal. Now, with that information, okay, it comes to another advanced technology of what we call as multiplex. Multiplex, okay, is what I call, it's quite advanced technology. Let me see if it works later on, because you can run yourself later. So multiplex is a technology using bits. Okay, so instead of using well like this, they convert this well to a bits. Okay, they convert the well to a bits. Very interesting, isn't it? So when you have well, it's going to be very limited. Okay, limited means the number of detection is very low. If it is in a bits, okay, the number of detection is very high. How is multiplex is more precise compared to the ELISA, or more interesting compared to the ELISA, is that in this one well, you can only detect one antigen or one antibody. But for multiplex, you can detect more than one antigen and antibody. It can be like hundreds, it can be 20, it can be 5. It depends. The more antigen or the more antibody that you want to detect, so the more what I call the cost it will be. But it is super cool. Okay, you just need a few sample, okay, from a patient, but you can detect many things: cytokines, antibody, whatever cytokines, interleukin 1, interleukin 4, okay. I don't think it's the kit is very expensive. All right. Now, an example method of an ELISA experiment. Before starting the work, read the kit instruction. Okay, you must do this. All right. Every kit will come with instruction. Read the kit the instruction of the kit. Different kit will have be different instruction. When you buy a new kit, then you have to think about, okay, I want to use this kit, and what will be the method for using this kit. All right. The next one, the problem that normally happen when I was doing ELISA as well. Okay, if I miss to label the 96 well plates first carefully using the correct color pen. Make sure that the plate is not wet. Okay, you write everything clearly. If you didn't do that, imagine you have 96 sample, you don't know which sample you are putting in. Okay, when the other problems happens during ELISA is also, you know, you have like lots, hundreds of samples. Imagine you're in the hospital, it's not only 3 patients comes in. Okay, it will be a lot of them. Okay, and then the doctor's pushing you, oh, I need the results today, can you give me the results today, otherwise the patient will die, whatever thing they want to say, it's up to them lah. But the thing is that you have to put that into consideration. Whatever things push you, you have to be very careful. Okay, put the label properly, make sure that you report the correct sample, okay, the correct data. Okay, if you do one mistake, I'm afraid that you might have to repeat the entire experiment. Whether you are going to pursue your PhD or master's, or you are going to enter into the diagnostic lab, it is up to you. But the thing is that make sure that when you run this ELISA, do it carefully, because the cost for one kit can reach to 2,000 ringgit per kit. It is only for 96, less than 96 samples. Okay, so please, be careful of this. Later on, when you are in the diagnostic lab, then you will understand what I'm saying. Okay, the sample must be, okay, when you run this experiment, okay, it must be in duplicate is best, but duplicate is enough. The reason why we do duplicate, because of the reason why we do this replicates, because we don't want, say for example, if one error, another is not, another 2 is okay, so we can accept the other 2. Imagine if you don't do any duplicate, you just have one well for one sample, what will happen? You may give negative results, you may give false result. Okay, so that is the reason why you need to do the duplicate, or the best is to do the triplicate. So imagine if it is 96 well plates, you have 96 well, and you have to get rid of like 12 of them for this for a standard curve and so on, and then you are going to do replicate. Say for example, duplicate only, you have so little number of samples, okay, and you have plenty of patients. Okay, that is why you have to remember, when you run this ELISA, you have to be very thorough, very careful. Okay, otherwise it's going to waste a lot of money to buy these consumables, especially when you are in the government hospital. Do not waste any single of the kit, because it comes from the taxpayer. you are responsible towards it. Okay, the quality control sample which is provided with the kit is treated as the test sample. So this is the control. Now, how do you determine the results? Okay, so the results is based on the absorption in nanometer and concentration. Okay, so you can imagine if I put a dot here. Okay, if I put a dot here, where is my annotate? Okay, annotate, and then draw. Okay, so if I put a dot here, okay, compared to dot here, okay, you will see the absorption over here, okay, will be less compared to the absorption at the top. So meaning that here your concentration nanogram per mil will be higher. Okay. Where is the other? Okay, I think that's all. Now I stop the annotate. Okay. Okay, that is how we normally do when it comes to the counting the results. Okay. As I said to you earlier, we have a standard curve. Imagine there is a standard straight line. Okay, so we will have an absorption over here. Okay, by reading the spectrophotometer, and then you can plot, and then you can pull it down, and then you can determine what will be the concentration. All right. Okay, let me try show you an example. So this is a standard curve line. You have a reading right over here, and then what you are going to do is that you are going to do like line here, okay, based on this NM absorption, okay, and then you will plot and you bring it down, and you will estimate that this is going to be X nanogram per mil. Okay, this is how we do in the test quantification of the, what do you call that, antibody, using the ELISA. But nowadays we don't have to do this anymore because everything is computerized. It's automatic. Okay, comes with the system. Okay, moving on to this one. Okay, this is what I just mentioned to you. There is a system. So you just have to do the standard curve, and then the standard curve, you are going to use the standard curve to give you the result. Okay, this is what I mentioned to you, absorption. You drag it over here, and then you pull it down. So it's similar to what you have done during your modern maths, if I'm not mistaken, during your SPM. All right, so this one I have mentioned to you. So this is additional reading. Okay, can we have like 5-minute breaks before we continue to flow cytometer?
Yeah.
Okay, let's have 5-minute breaks. We will meet again at 10:30, inshallah. Okay, Assalamualaikum warahmatullahi wabarakatuh. Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Let us continue our Allahumma salli discussion. Okay. Right. Now we go for flow cytometry. Okay, flow cytometry. Right. Okay. When we talk about flow cytometry, this is another advanced machine that we use to detect many things, including bacteria. Okay, we detect what I call the particles. Okay, very, very interesting. Even you can quantify the number of microbes. Okay, you can quantify the number of any kind of microbes using the machine, flow cytometer. So that's why I put here flow cytometer. Flow cytometer is a technique used to detect and measure physical and chemical characteristics of population of cells or particles. Okay, so it uses it's quite advanced machine. Okay, if I don't know whether there is a flow cytometer at the Kuliah of Allied Health, but there is a flow cytometer at CREAM. Okay, so the concept is that this machine uses laser. Okay, and there will be like a liquid. Okay, will go through the flow chamber. So this flow chamber we will direct, okay, the movement of the cell suspension. So you have several cells, okay, so it will be go through a single file. So from here, from the mix of them, it becomes like a single what do you call liquid, single, I would say, molecules, okay, particle cells will go pass through, okay, and once it reached to the laser, it will be bound, okay, to several site scatter. Okay, so each of these will have different dichroic mirror, which will read at different wavelength. Okay, so you have site scatter, you have F1, F2, F3. So all of these will give different wavelengths. Okay, and they will be detected by the fluorescent detector. And this information from here will be come together with the forward scatter, joined together, and gives you the results of what is the level of cells or particles, okay, or antibodies, whatever you want to detect using the flow cytometer. Okay, a very simple, very straightforward. It's quite expensive. The machine is very, very expensive, I would say that, but you can easily detect even up to the picogram, okay, pico level, okay, milligram, milli dah kecil dah, okay, nano, pico, kan, so up to that level. So a very, very super sensitive machine that based on the detection of bandwidth or detection of the what I call that, the laser. Okay, so it uses the technology of the laser in this case. Right. Whatever I just mentioned to you, I am summarizing into 5 principles. The first one is light scattered. Okay, this light scattered by the laser. Okay, and then the second one, it is specific fluorescent detection. Okay, the third one, it is hydrodynamically focused stream. So this is the hydrodynamic, right? And then the fourth, electrostatic particle separation. It's over here, the separation, and then it is a multivariate data analysis capability. So all the analysis will occur in the computer. Okay, so what you have to do at the end of the experiment, you just have the data ready and you just present, to the doctors or to anyone that feel that this result is important or necessary. All right. Now, let's talk about the principle. So the principle of flow cytometry again. We are talking about the light scattered. This is the source of the laser. So there will be the forward scatter detector, which I have shown to you earlier, the forward scatter detector, and there will be also a side scatter detector. So this on the side area, okay, which detect different wavelength, it is side scatter detector. Okay, so all of these are side scatter detectors. Okay, so we have four side scatter detectors together with the forward scatter, which give you the final and data analysis. Okay, so you already know the concept. Yeah. Now, similar diagram. Okay, what are the components? The most important what are the components that make a flow cytometer? Okay, it must have a fluid. Yeah. And the second one, it must have optics. Okay, when we talk about lasers and so on, it is optics. It must have a detectors. Okay, so you need to have a detector. You have lights, no detector, no point. Okay, last but not least, this involves electronics. So you have to understand, as a scientist, as a what they call biomedical experts, you are trained as a individual works in the lab, in a diagnostic lab. So we have two types of biomedic, if I'm not mistaken, when I was in UM. So we have biomedical medic, we have biomedical engineering. Okay, those who are working in the biomedical engineering, so they are the one who are doing this device. Okay, whereas for your biomedical medic in the Faculty of Medicine, UM, okay, so they are the one who work on the diagnostic. Anyway, at least for this time point, you should be able to understand that these are the major components when we talk about flow cytometer. If I put something else, then it is like, for example, I give you fuse, I give you what I call electricity. Yeah, but that is not the components. Okay, so the most important components are the four. I can give example as question as that. Right. So more details on the scatter, you can just browse it here. I've already given to you in the YouTube. Now, so you have the light scatter gating. So this is called the gating. You can detect neutrophils, you can detect monocytes and lymphocytes, everything from human white blood cell, from the human lymphocyte. From the lymphocyte, you can detect everything. Okay, so all of these, the higher intensity will give you different scatter projection. So this is high, this could be lymphocyte. Okay, the machine normally gives you what are the things mean. Okay, so you have the forward scatter, side scatter, forward scatter combined together in the machine. Machine will do the analysis, give you the result. Okay, this is the location, and you will detect. We will determine this is what, this is what, this is what. Yeah. Okay, more details about flow cytometry, I put it in the what I call in the YouTube link. All right. Because we have a few more minutes, I have another meeting at 11. I'm so sorry about that. Now, look at this. When we talk about what I call that flow cytometer, okay, flow cytometer, it is based on granularity, fluorescence, and size. So we have this. Okay, it is based on the size, I would say the most important. Okay, so because of the size, it will give you the location. It will give you more understanding of what are the cells that you are detecting in the particular sample. Okay, it is also using this what I call the technology of fluorescence, which we have also discussed in the what I call in Eliza, right? But for more details on immunofluorescence later on, inshallah, we will discuss this Friday. But the thing is that at least you have to understand in this machine, okay, in this technology, okay, we add a fluorochrome label antibody. Okay, so this fluorochrome label antibody, okay, together with the antigenic surface marker, okay, when we bind it together, if it is detecting, if it is bind to the specific antigen of interest, what will happen is that it will also give the same result as ELISA, detecting the color. And from this detection of the color, the computer will capture this color information and analyze it, which eventually you get how much or how many, what is the location also from the particular interest cells. Okay, so the fluorochromes that gives the colors here can be various, such as FITC, we call this as FITC, PE, PERCP, and APC. Okay, FITC stands for fluorescein isothiocyanide. Okay, PE phycoerythrin. Okay, PERCP pyridinine chlorophyll protein. APC will be allophycocyanin. Okay, so for the sake of exam, so you don't have to remember all these long names. Okay, if I ask you, give me the examples of fluorochromes, just give me FITC, PE, PERCP, and APC. That is more than enough. Okay. Now let's go to the application of flow cytometer. As I said to you, okay, this flow cytometer is very interesting because you can count the number of cells. You can count the number of monocyte, lymphocyte, lymphokine, so whatsoever cells that you want to count. Okay, even the number of bacteria will be easy for you to count as long as you use the correct fluorochromes. Okay, if you put the bacteria there without fluorochromes, it will elicit any here, and nothing there. Okay, so the next one, what is so interesting about this, it is also sorting from one white blood cell. It sorts into several. Okay, so this is also very interesting. Another interesting application for flow cytometer, it is also help in the detection of microorganisms. Here I put here determine cell characteristics and function. It's okay. It can also be the biomarker if you want to detect whether someone's having HIV, having certain diseases. So you just use flow cytometer, it will help you to detect, okay, whether this is potentially is having certain diseases or not. Okay, meaning that if you want to detect patients having COVID-19, you can also use flow cytometer. Okay, and also you can detect the protein from flow cytometer. Last but not least, you can also detect someone's having blood cancer or not from using the technology of flow cytometer. So this flow cytometer, I would like to give you Nila some activities. Okay, some activities. This is up to you. This is up to you. If you want to do it, then you can do it. If not, I do not force you to do. But I just want to see if you can like Google search around and create something like an experiment, a dummy experiment. How is the ELISA will look like? You can give an example of, okay, COVID-19 vaccine working or not working. Okay, it works or not. Okay, and then you make what I call the you isolate the saliva or blood or whatever you want, and then you run the ELISA. But the results you cannot put lah because this is what you didn't run the real experiment. But at least you know what will be the steps, step by step. Okay, what is going to do, what you have to do next, what is your what do you call that, the results of that you can predict. Okay, based on what you have read, going to COVID now and so on. So far, I just give an example. Okay, no harm, no force, does not give you extra marks or whatever. Okay, so this is only to increase your understanding, whether you really understand ELISA. Because flow cytometer is using, it does not have really so much complicated thingy to be explained, as long as you know it uses the technology of laser and detector. Okay, then it will be fair enough. Okay, later on, inshallah, when you are in the facilities, then you will go for details on how to do the what I call that analysis. Because flow cytometer, it will immediately give you the analysis, so you don't have to worry so much to do the tabulation of data, which I have shown to you when we do Eliza. Very, very straightforward. Like when you do Control-C, Control-V, you can choose copy or what do you call that, paste, just by searching something on the top of your Google, of your Word, Microsoft Word. Yeah. So that is my analogy. Okay. So I think I hope that is clear to everyone. So I stop my sharing session, my sharing here. But before I end my my lecture, my sharing, so as I said to you, I am not so keen to just giving lecture without listening to you guys. So I hope that is, if it is okay, to have the one that I give you in the end of the slide, you can submit it anytime. Okay, it is not your what do you call that, a must for you to do. Okay, you can just conteng saja, that is also fine. At least if you want to show to me, okay doktor, betul tak macam ni ke design dia, oh macam ni ke doktor buat eksperimen macam ni ke, just to confirm, just to make sure that what you learn here is something that is understandable or useful for you. So you can just create, design it and then show to me. I have already put group 1, group 2, group 3, group 4. If you have any groups, and then you can make it. If you want to do all by yourself, is also fine. Just send it to me to my email, and then I will comment if you want me to comment. Okay. Now for the next lecture, as I said to you, I don't want to go always giving lecture, because giving lecture so much is something that's very, very boring. I have been doing this for 5 years. Not 5 years, 4 years and a half. And then there is nothing new. Just a little bit information here and there. And now I would like to listen from my students, if that is possible. Okay, so we have another 2 topics, which is immunofluorescence and electrophoresis. Am I right, Nurhanis? Shakira, betul kan?
Betul.
Okay, so we have immunofluorescence and Allahumma salli ala Muhammad, electrophoresis. Okay, so that will be this Friday. That will be this Friday lah, betul lah, Friday. So Friday from, let me have a look to the timetable. So it's going to be Friday from 2 p.m. to 5 p.m. Okay, so since the class starts at 2, I think some of the brothers will be rushing for the Jumu'ah prayer. I know it's going to finish quite early because Zohar is also early. So can we start about 2:30? Okay, we give like 30 minutes break for the brothers. Is that okay?
Yes.
Yeah. So we start at 2:30, then we can finish Asar lah. By Asar, Azan Asar, we stop. Okay. If we need to finish at 5, then we will finish at 5. By default, we will finish by Asar. Azan Asar, stop, and then we dismiss the class. So I think that it will be my last. Next class will be my last class. This is my first class. All right, and then there will be few more sorry. Do we have any more lecture after this? Other than how many more lecture?
It's supposed to be on this Friday and next week, Tuesday. But for Tuesday next week, you're supposed to teach flow cytometer.
I thought flow cytometer together this week. I have to cover both. Okay lah. So if that is the case, do we have tutorial?
Tutorial.
We have quiz, I guess.
Oh, quiz.
Okay. So we have quiz. So if that is the case, I don't know if since I already covered the flow cytometer, too advanced ni. All right. So for Okay, for Friday, okay, for Friday, we will have, we are going to cover, what do you call that, flows, bukan flow, both lah, electrophoresis, okay, we will cover, okay, so on this Friday, we will cover electrophoresis, okay, and on the 23rd, which is on Wednesday, Tuesday, we will cover the immunofluorescence. Would that be okay? Then on Friday, we will have like 1 hour class. And on Tuesday, we will have another 1 hour class. So instead of having continuous 3 hours, we make it 1 hour, 1 hour. Would that be okay, Shakira, and all the members?
Yes.
Okay. So if I don't, if we didn't do the class on the 23rd, I think it is going to affect your attendance. So we make it for the sake of attendance, we have to do it. Okay. And then we will have it, what I call on electrophoresis will be on the 23rd. Okay, immunofluorescence will be on the 19th. Okay, regarding the electrophoresis and immunofluorescence. Okay, so how many groups do we have, Shakira? Do you have groups?
No, we do not have a group.
Okay, now can I make a group? Okay, so we have about 27 participants here. Okay, if I'm not mistaken, you have 29 students, can?
Yes.
Yeah. So with these 29 students, would it be, can we divide into 4 groups? Okay, we divide into 4 groups. Okay, 2 groups will be explaining on immunofluorescence. Another 2 groups will be explaining on electrophoresis. Okay, so the first 2 groups, okay, on immunofluorescence, okay, you will, since we are going to start our class, we are going to have 1 hour session instead of 3 hours. Then our class on Friday, we will start at 3 p.m. until 4 p.m. inshallah. And then the class for the 23rd will confirm with you later on Friday. Okay, so for the class on Wednesday, the class on Friday, this Friday on the 19th, so we will start at 3 o'clock. We will start with the 2 groups, okay? Any of the lucky groups, okay? You can explain what is what is immunofluorescence? What are the function of immunofluorescence? What are the example of the immunofluorescence? And what do you want to know more about immunofluorescence? If you have any questions that you would like to ask, you would like us to search during the class, okay? Anything you want to share with us, all right? So from your lecture, okay, I will verify it, and your lecture will be used for me to make the final exam question. So make sure that your information is correct. Then this information will be used for your final exam. Okay. Now there should be no issue. Okay, I should have like 10 of you should be very, very excellent in immunofluorescence, and then another 10 of you will be very, very excellent in electrophoresis. So the questions will also come later on following our discussion after the lecture on this Friday and Wednesday. I hope that is okay, Shakira, Sharifah, and all. Would that be okay?
Yes.
Okay, so you have okay, so the presentation will be at 3 o'clock. Okay, I think this is going to be online or face-to-face?
Online.
Online, okay. So we are going to make it online. How long is the presentation? So we will have this presentation maximum of half an hour for each group. Okay. Okay, so meaning that we will have like 20 minutes presentation, 10 minutes of Q&A, and then another group will be the same thing. Okay, so the first this Friday will be on immunofluorescence, and the other two will be on the 23rd, which is on the electrophoresis. So I think it's going to be difficult for you to distribute the groups. So can I distribute the groups now?
Yes.
Okay.
Sure.
Okay. So we have 27. Okay, I just call a name lah. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Okay, based on your attendance, attendance, mana attendance? Okay, I go to your attendance, my attendance list. Okay, masuk. Okay, those yang Shakira, make sure that those who are not able to join us, just give him or her any groups. Okay, so far the one that log in is 27. Okay, 27. Okay, 27 divide by 4. Okay, so we will have 6 and 7 groups. Okay. Hanis Fatini group 1, Shahira group 1, Shafiqah group 1. Okay, Shafiqah, everyone can hear me, right?
Yes.
Yes.
Okay. Anis will be group 1. Okay. Shafiqah Yusriha, okay, group 1, Raihana binti Ismail, Intan Raihana group binti Ismail, okay, group 1 juga. Okay. And then untuk group 2, Fadilah binti Muhammad Fauzi, Masturina binti Fazid Osman, Najiyah binti Kamaluddin, Hayati binti Marzuki. Berapa dah tadi 1, 2, 3, 4, 5 kan?
4 doktor.
4 4, and then Nabhan Lutfi 5, Nur Nadia binti Fatihi 6. Okay. Next group number 3, Nur Akilah binti Ismail number 1, Nurul Raudah number 2, Haizatul Umiyah number 3, Sharifah Dora number 4, Nur Farihah binti Ahmad Radauddin 5, Nurul Ain 6, Nik Nur Alia 7. Okay, Nik Nur Alia 7. And then the last group, Nur Farhana binti Muhammad Rosli group 1. Group 4. Okay, number 1. And then Nadia Nur Shifa 2, Mizan Kamalina 3, Nur Farhanah Aqila 4, Nur Azah binti Muhammad Idris 5, Sharifah Nadia binti Said Ahmad 6, Nur Shakira binti Muhammad Ansar 7, and last 7. Okay, so Eva Nazura binti Nazri Shah go to group number 1. Okay, you go to group number 1, Eva Nazura binti Nazri Shah. If you ask me again, the name I don't remember lah. So the rest you can just jumble up as long as you have 6 to 7. Okay, I leave to your creativity whether you want to use what do you call that, Canva ke, PowerPoint ke, whatever, it's up to you. Okay, as long as on Friday, group number 1 and group number 2 will be presenting on what do you call that, will be presenting on immunofluorescence. Okay, group number 3 and number 4 will be presenting on electrophoresis on Wednesday. So will be 1 hour session. Okay, I think that's all for our discussion today. Anis, Fatini, okay?
Okay, doktor.
Okay. Raudah, so far so good?
Inshallah, so far so good.
Okay, inshallah. Okay, remember, everyone, you are my colleagues, I would say. If you encounter any problems, if you have any problems, okay, not only on this flow cytometry, flow cytometry, what else? Dia punya tadi yang awal tadi, Eliza, spectrophotometer, spektro ke? Bukan spektro. Panggil apa tadi lupa dah. Electrophoresis, electrophoresis, or even on immunofluorescence, anything lah, inshallah. Okay, if you have any problems, okay, just contact me. I will try to answer to you the best I could. Yeah. So there is no question is stupid questions. Okay, all questions are good questions. Even a simple question you think is a simple question, sometimes it's not a simple question. It can be very hard question. Maybe simple to me, but it could be difficult to you. So, but now, my dear students, remember, okay, you are similar to what I am, what I was before. Okay, know nothing. I start everything from zero. So if you have any problems, okay, just let me know, inshallah. I will try to assist accordingly. So let me open to everyone again. If there is any more, if not, then I can go to my meeting because I'm supposed to have a meeting with Prof. Hafiz and Deputy Director at 11, but class is far more important. Okay, can I just go through one by one? Alia, okay, Alia Aziz. Okay, Akilah Suhaimi.
Okay, doktor.
Okay. Okay, Fadilah Fauzi.
Okay, doktor.
Okay, Farhana Rosli.
Okay, doktor.
Farhana, so far okay? Okay. Haizatul Umiyah.
Okay, doktor.
Okay, so majority dah lah tu. Okay, insyaAllah. If everyone is okay, no question. Last lah, Nabhan. Boleh
Boleh doktor, alhamdulillah boleh.
Nabhan seorang je ke yang lelaki? Ada lagi tak lelaki lain?
Dalam diagnostik saya seorang. Kalau
Seorang kita je. Ada saya jadi dah tambah 2 orang lelaki. Nasib baik lecturer semua lelaki ya yang ajar subjek ni. Tak adalah nasib baik. Nabhan seorang saja lelaki diagnostik.
seorang saja.
Yang lain-lain lelaki pergi mana?
Yang 2 orang lagi research.
So total ada 3 je lah lelaki.
Betul doktor, 3.
Berapa banyak perempuan Syakira?
28.
28 sisters and we have 3 brothers. Oh, mana pergi habis semua Nabhan kita ni? Tak apalah, mungkin ada kuliah. Okay, so I think that's all. Thank you very much. Let us end our discussion today with the recitation of Tasbih Kafarah and Suratul Asr. Subhanakallahumma wa bihamdika asyhadu an la ilaha illa anta astaghfiruka wa atubu ilaik. Wal 'asri innal insana lafi khusr illa alladhina amanu wa 'amilus solihat wa tawassaw bil haqq wa tawassaw bis sabr. Insyaallah jumpa lagi Jumaat ni. Assalamualaikum warahmatullahi wabarakatuh. Jumpa lagi semua.
Thank you doktor.
Sama-sama.
Thank you, doktor.
Thank you, doktor.
Thank you, doktor.
Thank you, doktor.